This Week in Parasitism is brought to you by the American Society for Microbiology at microbeworld.org/TWiP. This Week in Parasitism, episode number 9. Hey everybody, I'm Vincent Racaniello, and with me today is a glowing Dickson Despommier.
Hey Vince.
How's it been, Dickson?
It's been great.
Here you've been in the Canaries.
I have been in the Canaries.
And what language do they speak there?
Tweeter.
Tweeter.
They tweet a lot.
Tweet. Do they have parasites in the Canaries?
That's a good question, actually. They're 250 kilometers off the coast of North Africa. And while we were there for a week at a sustainability meeting, where we met, by the way, people from the Galapagos also, because there are bunches of communities throughout the world that live as island communities.
Isn't that where Chuck was?
Chuck. Oh, Chuck, that Chuck. You mean Charlie. He was there several times.
So the Canaries are close to the Galapagos?
No. They're equivalent to the Galapagos,
yes.
But they're off the coast of Africa, whereas the Galapagos are off the coast of Ecuador in South America, right? But they were all formed by volcanic activity. So all I saw all day long, all night long, were peaks of volcanoes, just everywhere. It was like walking on Mars at some point. Big lava flows. Very, very primeval stuff. A lot of the islands are built up, but some of them are just absolutely gorgeous. The sides of the islands that face Africa are as dry as the Sahara Desert itself. The sides of the islands that face the Atlantic Ocean on the far side are verdant, beautiful, gorgeous, almost like rainforest quality. It's where Christopher Columbus first navigated to go to the New World, because the trade winds, and they're called trade winds, actually allows a sailor to go from Spain along the north coast of Africa, where they pick up these sahals, or these shiracos as they're called, and it blows them all the way across the Atlantic, and then they pick up the Gulf Stream to come all the way back. So it's a big circle. And that's how Christopher Columbus actually completed his 4 voyages. And one of his residences is in Gran Canary Island, and so we went to visit that, and we saw a lot of his artifacts and models of his ships, and the kids. It's a great experience. I highly recommend it. And plus, we work at a university that bears his name.
Columbia University.
This is right.
Is that right?
That's exactly right.
Is it somehow derived from Columbus? Yes.
It is. It's directly derived from Columbus.
I know it was founded as King's College initially, right?
Yes.
And then it was changed to Columbia at some point.
As soon as the revolution occurred.
Yeah. But do they have tapeworms?
But do they have tapeworms?
Not just tapeworms, parasites. Do they have tapeworms?
There are parasites everywhere, Vince.
Malaria?
I would assume so. I didn't hear much about it, but I would assume so.
Toxoplasma?
Oh, toxo is every place. So parasites are wherever you find hosts.
Of course.
Can't have a parasite without a host.
It keeps the population in check.
That it does.
Now, Dickson, we have talked so far in this course.
Yes, right, this course, please. Listeners, don't take this serious.
Talked about a lot of tapeworms. We've talked about trichinellas, which are round, nematode-like worms.
They are not like just nematode-like. They are nematodes.
They are nematodes.
Yes.
And we talked about tapeworms, which are segmented.
That's correct.
They are, what's the word for them again? Wait, I'll look it up here. See, the thing about being a scientist, cestodes.
Yes.
I have to think of cesspool. The cestodes are the tapeworms, fish and beef and pork.
And dog.
And then the nematodes were the trichinellas.
That's right.
And that's what we've done so far. We've had a few general lectures, but we haven't spoken about any protozoan parasites.
We have not.
Only briefly.
Correct.
Is it possible that we will today?
It is very possible that we will.
Or are you going to read a story?
I know I did that last time.
A number of people wrote in saying they liked it.
Oh, good.
But they did say that nothing beats the repartee.
I guess.
You know what that means,
Yeah,
right?
I do.
You and I talking.
That's exactly right. The banter.
But they did like it.
The banter.
So what's on the menu for today, Dickson?
Well, that's a good question, Vince, because that's exactly how you catch most of these parasites. If you're not looking carefully at what you're eating, perhaps something will be eating you instead. We could continue the discussion with worms because we didn't discuss the schistosomes yet, or the flukes. Or we could hold up on that and go into the next big section of parasitism with regards to the world problems as they exist, and that is to discuss protozoans, or single-cell organisms. And the top of that list, not the most frequently occurring, but the one that causes the most health issues, is malaria by any stretch of the imagination. Malaria leads the world in terms of human suffering.
Now, you said not at the top of the list. Why did you say that?
Because there is another parasite called Toxoplasma gondii.
It's even more
There are
prevalent? Are you serious?
billions and billions of people.
Wait, now I have a table here, Dickson, of worldwide mortality due to infectious diseases.
Mortality is different.
That's different. So the toxo doesn't mortalate you.
Does not. In fact, it's one of the best parasites in the world, and we'll probably take 2 sessions to talk about toxo.
I see, because here on the deaths, we have lower respiratory tract infections are the number 1. Number 2, what would you guess?
Malaria.
No, AIDS.
Okay.
Number 3 is diarrheal disease.
Oh, sure.
Number 4 is tuberculosis.
Okay.
And then we have malaria.
Right.
And surprisingly, right after malaria is measles.
Which shouldn't happen, right? Because we have a vaccine.
We have a vaccine, but a good part of the world doesn't get it.
That's right.
And the parts of the world that don't get it, those children are malnourished and makes measles much more severe.
Here.
But, so malaria is number 5. Well, and that's a protozoan.
That's mortality. It's got an extremely low mortality rate based on the
malaria.
number of cases.
Well, a good parasite would do that, right?
A good parasite would do that.
If I have learned anything from the tapeworms, it's that they want to curl up inside you and live as long as you do.
Hey, exactly.
And then when you die, someone else will pick them up.
This is true.
One question I have had arise in my brain is, when we talk about viruses, we always say most of the new infections are zoonoses. They are infections coming from animals.
That's right.
Where do parasitic infections come from? Now, we know today where our infections come from, but years ago, where did they originate from an animal parasite?
During our evolution?
Yes.
Well, that's a wonderful question because it's so wide open that it varies depending on the organism that you discuss. You're asking me where these diseases came from in terms of our evolution or their evolution, and you have to speak about both of them in order to know the right answer to this question. And the answer is that before there were humans, there were parasites.
Correct.
All of these parasites had other hosts, other humanoids first, or hominids, I guess they're now referred to by the anthropologists. So hominids of all kinds: Homo erectus, Homo habilis, Homo, Australopithecus europus. All of these other groups of hominids that gave rise to us, carried with them the inheritance of their parasitisms from even more primitive forebears. And some of these parasites can cross lines and infect many different mammal species, and others are absolutely restricted to their hosts, to the specific species. Okay, and we've discussed a few of these earlier.
Well, a lot of the tapeworms have multiple hosts.
That's correct.
But they have different stages and different hosts.
This is right.
So presumably the precursors that before, let's say a pork tapeworm, where we're infected by the larval stage, correct?
And the adult.
And the adult, not the eggs, though.
No.
Typically.
Well, no. The eggs, if you swallow those eggs, they will hatch and develop to juvenile stages in your body. They will. The pork tapeworm will do that.
But we typically don't ingest the eggs.
But the beef, but whenever we do, though, we get some medical difficulties, remember?
Yes.
Whereas with the beef tapeworm,
which
looks almost identical in terms of the adult worms, and the eggs look identical in terms of their morphology. If we ingest those eggs, nothing happens to us.
So the point is we have 2 tapeworms. We had beef human and pork human. Before the humans, what was the other host? It was some other animal?
Some other hominid.
Who ate those guys, basically.
Sure, that's right.
Right. Oh, some other hominid, yes.
Right, and then you go back before that, some other primate, and before that, some other pre-primate, and before that, that's how these things evolve. If you looked at the Trichinella lesson that we learned, that it infects all mammals. There isn't a single mammal out there that isn't susceptible to Trichinella, which is remarkable. But if you look at the host range of Trichinella with regards to mammals versus birds versus reptiles versus amphibians, There's only one species of Trichinella, Trichinella pseudospiralis, which infects humans and birds. And all of the other species of Trichinella either infects, or they infect only warm-blooded mammals, basically.
All right. So this is probably a question for an entire session.
It is.
But
Host specificity is a huge issue in infectious diseases. And it begs the question, I mean, we've talked about this in TWiP also with regards to influenza virus and other viruses as well, rabies, for instance. Those are all, they cross over from one species of host to another. And in the host that they're most adapted to, they cause no problems. In the host that they're poorly adapted to, they cause huge problems. Okay. So the same is true here.
Yeah.
Okay.
We haven't really talked about it.
We have not, but we've hinted at it all along by saying who gets it and who doesn't.
Now, new viral infections are always emerging.
Of course.
Are new parasitic infections also emerging?
You bet. And in fact, today,
yes,
we're about to discuss malaria. And for many, many, many years, it was thought that there were 4 major species of malaria that infected humans.
I see them right here in your textbook, huh?
They're right there in the textbook, and exactly. There's a couple of odd ones that occasionally infect people. But we now know that there's a variant of one of the 4 species listed there in the table that has been newly discovered in West Africa. And so we now have another one to add to our list, perhaps of a new species of malaria that infects humans. A variant of Ovale.
It's probably safe to say that new parasitic diseases don't emerge as frequently as new viral diseases, because viruses evolve so much more rapidly.
Yeah, they have a rapid replication cycle, so there's more room for variation and more carrying forward of traits that you'd probably select for. So, so malaria parasites, let's talk about the group of parasites called malaria. Okay, because remember when we first talked about who gets it and how much disease it causes, I said you have to define it as how much infection, and then what is the mortality rate in that infection. Okay, so the most frequently occurring parasitic infection that's a eukaryotic parasite is Toxoplasma gondii, by far the most frequently occurring parasitic infection in humans. Even more frequent than pinworm, which is a very harmless kind of a nematode that infects mostly children. And that's found throughout the world. This parasite is found even more frequently throughout the world. And it's acquired in 2 different ways. One is by ingesting infected meat, similar to Trichinella. And the other is through contamination of the stages that are produced in the cat or in felines as a result of they're the definitive host for this parasite, Toxoplasma. Well, the reason why I'm even mentioning it is because it's related to malarial parasites. Okay?
How so? How are they related?
They're both part of a large group of parasites called the apicomplexa.
Oh, dear.
When you do the
How do you spell that?
No, come on, Vince. You've got stuff like that in virology that confuses the hell out of me also.
How many different genome types, Dickson?
Oh, many. 7 in viruses. But
how do you spell apicomplexa?
Apicomplexa.
It's a group which includes the malaria and...
Toxoplasma.
Anything else?
Yes, Cryptosporidium, which is another frequently occurring protozoan infection throughout the world, which induces diarrheal disease, by the way. So you throw those 3 groups together, and what you've done is you've encompassed maybe 80% of the mammalian life on Earth that's susceptible to these infections. It's quite amazing, actually. So in terms of morbidity, though, the disease-producing ability of an organism, malaria is by far the champion of all the world. Of all the eukaryotic parasites, in terms of frequency and disease, it is a champion at this, okay? And yet at the same time, because it infects so many people, perhaps more than 2 or 3 billion people at any moment are infected with malaria.
Is that right?
This is correct.
Wow.
It only kills about 1% of those people. Yet, if I told you that 3 billion people are infected, what is the mortality rate? It's 1%. And how many people
does that? That's a lot of people.
That's a lot of people. So many people have died over the last 100 years that now equals the size of the population of the United States.
It says here that there are each year 350 to 500 million new cases of malaria, killing between 1 and 3 million people.
That's right.
Most of which are young kids in sub-Saharan Africa.
Correct. Which is just across the way from where I just came from. So you're asking me if there are parasites on the Canary Islands. I can't imagine that some mosquitoes don't blow over across that 200-mile stretch.
200 miles. It's about the same as Madagascar from Africa. Is that right?
That's correct.
All right. So are there known cases of malaria on the Canary Islands?
I didn't find that out while I was there. I actually don't know, but we could probably go to the morbidity mortality reports and see.
Do we have malaria in the U.S.?
We used to have malaria
Right.
in the U.S. We don't have any what they would call autochthonous malaria any longer. And the reasons are multiple, and we can get involved with that.
What does autochthonous mean?
That means it comes from within the environment.
Like endemic.
Yeah, that's right.
So yellow fever was imported
That's right.
to the U.S.
That's right.
So that's not autochthonous.
No, not now, but it used to be. We used to have tons of yellow fever in New York City.
No, because the mosquitoes were brought here at some point, where an infected person came and imported the disease. It became established. It went as far north as Boston, I believe,
that's
right?
right. That's
and then we eradicated it, right?
Well, look at the West Nile virus. I mean, we brought the virus to the New World, but we already had the mosquito that carries it already here. Which, by the way, came from the African continent as well.
So the burden is sub-Saharan Africa. Where else are there major Asia?
Asia, sure. Wherever tropical and subtropical world has malaria.
So Russia doesn't have malaria.
Southern Russia, yes.
Southern Russia, yeah.
Sure. South Korea has malaria, believe it or not.
Italy?
Italy used to have malaria. In fact, we'll talk about the history, and Italy figures prominently in the discovery of malaria.
All right, so let's go.
Okay. So, malaria. It's a protozoan infection.
What does that mean, a protozoan?
A protozoan is a classification of animal life which revolves around the concept that it has one nucleus, usually, although some protozoans have multiple nuclei. Like, for instance, some of the free-living ciliate protozoans, like euplotes, for instance, or paramecium. You can find multiple nuclei inside. Usually there's one macronucleus and several or many micronuclei, and the genetics of these organisms are bizarre. Just leave it at that. People are still trying to figure it all out. It's kind of amazing stuff how these organisms carry out their lives.
So does that mean one cell?
One cell, one nucleus.
Protozoan.
Protozoan. So it precedes a zoan. Well, actually, we have another category of it's called metazoans, and those are the multiple-celled organisms. So
you and I are metazoans.
And so are nematodes, and so are cestodes, and so are trematodes. So those are multiple-celled organisms that infect other multiple-celled organisms.
All right.
So, but malaria is a single-cell organism, and it is a vector-borne disease. We've discussed vector-borne diseases before in our TWiP lectures, in our TWiP presentations, and this happens to be one of the very first organisms that was shown to be carried by an invertebrate. Do you know the one that was first shown to be carried by an invertebrate?
This is before viruses, right?
This is before viruses, that's correct.
No, I don't, Dickson.
Well, the first vector-borne disease ever discovered was discovered by Theobald Smith. Does that name sound familiar?
Yeah, there's a building at Rockefeller named after him.
Is that right? Well, how coincidental is that? It happens to be that
worked there.
Theobald Smith actually did work for the Rockefeller medical. I guess I'm trying to figure out what the original name for the Rockefeller
Medical Institute.
I think that's right. And half of it was in Princeton, and the other half was in New York City. In fact, I knew someone who lived in Princeton, who commuted from Princeton to the Rockefeller University, as it was then known as when I was there.
And now it's the Rockefeller University.
That's right. And they no longer have an affiliation in Princeton.
Rockefeller Institute for Medical Research, founded in 1901 by John D. Rockefeller.
Correct.
All right, so Theobald Smith.
Theobald Smith was an infectious disease microbiologist working at the turn of the century, and preceded the discovery by several eminent parasitologists with regards to their discoveries of the transmission of diseases. He discovered that Texas cattle fever, Babesia, which is another protozoan infection, was carried by ticks, by the Lone Star tick. And Vince has got it punched up on the computer screen, and there he is, Theobald Smith, a very distinguished-looking gentleman, a very mild-mannered individual, who cleverly figured out that this horrible infectious disease that was killing cattle all over the place was transmitted by ticks, and proved it by doing Koch's postulates, which was remarkable.
And that led us to be able to control it at some point, right?
Well, if you can control the ticks, you can control the infection, but controlling ticks is very difficult. You know how they do it in Africa? The Maasai have a great way of controlling ticks for their cattle. They burn off the understory every now and then.
The understory?
Yeah, you know, the grass.
The grass.
The understory of the
They
veldt.
burn it off to get rid
They
of the
burn
ticks.
it, and not only does it fertilize the ground again by putting the inorganic material back to the soil, but it also kills off the larvae and adult ticks that are in the grass waiting to go onto another host. So that's a kind of a naturalistic tick control.
So that was the first vector-borne
the very first
disease.
one to ever be shown to be transmitted by an invertebrate from one mammal to another.
And then after that came malaria?
That's correct.
Who did that?
Sir Ronald Ross, working in India.
India?
And some Italians working near Rome. And it was interesting because the history of this is rich with, let's say, all of the foibles that accompany scientific discovery, no matter when. It involves personalities and places that are exotic, like the center part of India.
Do you know some of these stories?
I know them all.
You know, our listeners love your stories.
Well, you can tell that I like telling them, so, that's probably why I enjoy teaching so much.
Yeah.
So it's it's a much longer history than we've got time for here, but...
We have forever. Not today, but, just...
Well, Sir Ronald Ross was a member of the British medical team working in India.
What rough year are we talking about here?
Back at the turn of the century, somewhere in the 1890s.
All right, so Koch had already established the infectious basis of disease.
There's a question. Bacteria were now known about, and lots of other diseases, lots of parasites were discovered and described during this era also. It was a golden age of discovery of infections.
All right.
So Ronald Ross was working for the British Medical Service, and he was assigned to a particular place in India, and had lots of time on his hands, decided to conduct some of his own research. And so he wanted to look into malaria. He thought
malaria was already known as a clinical entity.
Oh, big time.
For many, many years.
Killing millions of Indians, of course, in India, and also many British settlers.
Yeah, in your book it says we've known about it for at least 50,000 years.
Oh, it's an old disease.
5th century BC, so we can tell. It says here, Hippocrates was able to differentiate the different fevers.
That is correct.
Oh, okay. That's what I wanted to know, how old it was.
It's pretty old.
And
but that's not 50,000 years ago.
I'm sorry, yes, that's even
5,000 years ago.
Why did I say 50,000?
I don't know.
Why didn't you correct me? Why didn't you smack me?
I just did. I do these in gentle ways, Vince.
I don't know where I found 50,000.
I don't either.
Wait, so 5th century BC is 5,000 BC.
That's right.
Plus another 2,000. So that's 7,000.
It's a long time ago.
Do you think, just speculating, that it might have been around 50,000 years ago in some other form?
Vince, I know it was around in that time because the human population bottlenecked at one point. Meaning that at one point in our evolutionary history, there might have been as many as 400,000 individuals.
Yes.
That's not a lot of people.
No.
And what do you think narrowed us down that way?
A genetic bottleneck. Malaria? Yeah.
We could have been eliminated by an epidemic of malaria. It could have killed everybody that was left. But where did we evolve?
What part of the world? Africa.
Yeah, right? What part of Africa? East Africa.
Near Egypt.
Well, below that. Actually, it was along the Serengeti Plain in a place called Old Divey Gorge. But there were other places that we might have evolved
That's where we have most of the evidence, right?
And that's where we have most of the evidence. That's where we've looked, right? Because we haven't looked everywhere. But we seem to have been able to date our evolutionary history best by looking in the East African region. And, of course, that's a hotbed for malarial transmission today. And we have to assume that it always was.
Sure.
So, we didn't know what caused it. We didn't know how we caught it, and we didn't know how to prevent it. So Sir Ronald Ross in that case, just Ronald Ross now, working as a young physician in India, decides to do some experiments on his own. So what do you think he chose to work on?
Sounds like malaria.
Yeah, malaria, yes, but there's many different kinds, you see.
Uh-huh.
Not just human malaria. So it's not an acceptable way of conducting research. I mean, it's very difficult to conduct research on humans. So he decided to conduct research on canaries, birds.
Canaries get malaria?
They sure do. They do. In fact, they do. So Ronald Ross systematically eliminated every single species of mosquito that could transmit anything to canaries except the one that did.
So it had been known that canaries naturally acquired malaria? How did they know it was malaria compared to human malaria?
It's interesting you should ask that, Vince, because there's lots of other history behind all of this. For instance, there's the Romanovsky stains, which preceded all of this, invented in Russia by a Russian physiologist. And he could now use this staining technique to look at cells. If you go just a little before that, Robert Hooke was describing cells in cork. So not in Cork, Ireland.
Yeah, the corks in your bottle, yeah.
That's right, in the ones that he emptied, I'm sure, before looking at the cellular composition of each. So Robert Hooke says, let there be cells.
Right.
Romanovsky says, I can't see them. And Mechnikov says, if I could see them, I bet you I could describe phagocytosis. Well, of course, all of that came together like perfect storm of science. And you had Romanovsky stains facilitating the development of the microscope, which was, of course, under development at the same time. So here was young Sir Ronald Ross sitting at his laboratory bench, now with all the tools necessary to both transmit the infection from canary to canary, but also to see the parasites. There were other people involved in this too. A man by the name of Laveran actually watched the parasites mate. There were plenty of other things because they have sex. There's male and female protozoans, just like you have with other things. Some are not male and female, but this one in particular happens to have a male and female side to things. So there is a way for recombination to occur, and lots of genetics involved in all of this. So Ronald Ross said, I wonder how canaries catch their malaria. Because if we can find that out, we might be able to decide how people catch it.
So he went through all the mosquitoes.
Yeah,
Why did he suspect mosquitoes?
Good question. Well, because ticks had been shown to transmit babesiosis, which was related a little bit to malaria, because the Romanovsky stains allowed you to see the babesia parasites. Well, what cells in the body do the babesia parasites infect? Red blood cells.
Same cells?
Same cells. So here's a parasite that looks like malaria. It's transmitted, but you know what? These little birds, they don't have ticks. There's no tick. You can't find ticks. They have lice. They don't have ticks. But what do they have? They have mosquitoes that are always biting them. So Sir Ronald Ross probably went through lice first, didn't even record the results. He couldn't find any associations at all, and then said, it can't be a tick because they don't have ticks, but yet they still catch malaria. If it's a vector-borne disease, he never used that term, of course, but he says if it's an invertebrate transmitting it, it's probably going to be a mosquito, because look at what happens. A mosquito bites this one, and bites this one, and bites. So he logically figured out how to conduct the research. And then, much to his surprise, I'm sure, he proved beyond a shadow of a doubt that a mosquito can transmit bird malaria from one bird to another.
How would he have done that? Would he have taken an infected bird and allow mosquitoes to feed on them?
Just like the Sir Walter Reed experiment for the transmission of yellow fever. It was done exactly the same way, except that was done with humans, and this was done with birds.
Okay, good. What kind of mosquitoes did he...
He used all kinds. Now, you should ask me another question then.
What kind of malaria did he get?
How many different kinds of mosquitoes are there?
That can transmit this, or just are there, period?
Just are there, period. So Sir Ronald Ross, Ronnie, we'll just call him Ronnie, looks around and says, where do I start? I mean,
so even then they knew how to speciate mosquitoes.
Well, there was a burgeoning field of what they call now medical entomology. And medical entomology had its own series of heroes also, and heroines, I might add, by the way. So it became obvious that there wasn't just one species of mosquito, because otherwise you would have been able to tell what was transmitting yellow fever, right?
Sure.
So yellow fever was already shown to be transmitted by mosquito.
At this point already?
Well,
not quite, no, because we're talking about 18...
because the Panama Canal had not yet been started.
Right, not until after 1900.
That's right. So
do that.
This probably gave them a hint as to the fact that it might be a mosquito-borne infection as well. But so Ronald Ross looks around and says, how many different kind of mosquitoes are there? Each one has a different breeding site. Each one prefers certain hosts over others. Host preferences for feeding mosquitoes. How
So we knew this back then?
No. He had to find all of this out by himself.
So what, he went and trapped mosquitoes? He could differentiate them?
He didn't even have to.
How?
He could do that. He could also go to the ponds.
Yes.
And take the larval stages of the mosquitoes that were not infected and raise them in captivity.
Very good.
And then feed them on birds to see whether they caught the parasites. Because the guys that had done the work in the mosquito stages had already published their results.
Very good.
Okay.
Makes sense.
So, in fact, I'm leading up to an interesting point here because eventually they ended up awarding Nobel Prizes for all of this work.
Mm-hmm.
And he got one of them.
No, it sounds like he should have.
He should have, is right.
Yeah.
And the guy who discovered the sexual matings of the mosquitoes, he got one also. Mm-hmm. So, Ronald Ross finally decides that it's this mosquito species that transmits Canary 1 to Canary 2 to Canary 3.
What mosquito was that?
It was a Culex mosquito. Okay. Culex, why don't you ask me another question, Hans?
How many mosquito species are there?
Thank you. Well, actually, there are two big groups.
Uh-huh.
There's the culicine and the anopheline groups, okay? And you can clearly differentiate them. They're quite easy to tell apart, right? How do you do that? It's how they land on a surface as an adult. The culicine mosquitoes land absolutely parallel to the wall, and the anopheline mosquitoes land at an angle. Yeah. So when you're alone in your hotel room at some tropical spot, and you're wondering whether it's possible for you to catch malaria when you turn the lights out and go to sleep and you're as I did at the Canary Islands just this last week. So the trick is to sleep with the lights on, by the way. The mosquitoes don't come around that way. That's a trick.
Yeah, but you don't go to sleep either, Dickson.
Yeah, but you have to because you're tired. Okay, so you look on the walls,
yes,
all right, and in fact, I've been in some hotels that say, please don't disturb the geckos. They're eating mosquitoes and protecting you. Mm-hmm. I've actually seen that. It's a little card that says, don't disturb the geckos. They're here to help you. So here are these mosquitoes. They either land parallel to the wall or at an angle.
Culex is parallel. Anopheles
is at an angle.
That's there correct. You go. Anopheles angle, A.
See?
That's
he's
how
got a mnemonic already for this thing. Way to go, Vince. Don't worry. Next time I'll ask you again, and I hope you get it right, because if you don't, you'll ask me about the 7 different genomic arrangements that David Baltimore invented.
How long can that last?
Right.
Go ahead.
So here we have Sir Ronald Ross having to differentiate between anopheline and culicine mosquitoes first.
Okay.
All right. And no matter which anopheline mosquito he tried, which species, none of them succeeded in transmitting the disease from a diseased canary to a non-diseased canary. None. But the culicine mosquitoes had another multiple choice question underneath that. Is it Aedes, which is a treehole breeding mosquito, which is the one we all know about for carrying yellow fever, or is it the most common mosquito found throughout India, the Culex quinquefasciatus?
So under the culicine mosquitoes, you can have Culex species or anopheline.
Aedes.
Aedes.
Aedes, that's correct.
And then the anopheline separate. Is that just the only species in that group?
Yes, anopheline. Well, there are other
minor ones.
Other minor ones, but we'll just stick with those.
So Albopictus is a culicine mosquito.
It is.
All right.
It is.
Because it's a Aedes.
Aedes
Albopictus.
That's
okay.
Correct.
Are these both kinds are obviously in the U.S.
Sure.
Right? They're globally distributed.
There are location-specific species, and there are generic location species.
If I saw one buzzing around this room now, Dickson, what was it likely to be?
It's likely to be a Culex mosquito because they're called the northern house mosquito.
Okay, so most of the kind we get in this region, the northeast of the U.S., would be a Culex.
And they're peridomestic, meaning that they love living near people, and they're the ones that are primarily responsible for transmitting West Nile virus.
All right.
So that same mosquito.
Let's review. I've just forgotten what you said. The whole thing I've forgotten.
Oh, my
He found it was a culicine mosquito.
Yep, because none of the anophelines spread the disease from the diseased.
Did not. And then we have Culex and Aedes.
And it was the Culex.
It was the Culex group species. Okay.
The most commonly occurring mosquito in India transmitted bird malaria from bird to bird. That's what he discovered. He published his papers. He then was promoted to the editor-in-chief for the British Medical Journal and returned to England to become famous. In fact, he was awarded a Nobel Prize, as I mentioned.
For discovering canary.
That's right. But in the meantime, there's this other group, you see, working in Italy.
Yes.
Big Nami's group. Big Nami, okay? And Big Nami is reading these papers coming out, and of course they were published in the British Medical Journal, right? And one of them says, malaria is transmitted to animals by mosquitoes. What a big news that is.
Mm-hmm.
Wow.
The animal being the canary.
Wow. What a big piece of information.
But did they think at that point, did they say, this must mean that human malaria is transmitted by mosquitoes?
Well, they said this is good for birds. It's got to be good for people too. So, by the way, where does the word malaria come from? You've got an Italian surname.
Male means, a disease, a sickness, just like malediction,
That's right. Or malatesta.
Malatesta, a headache.
That's right.
All right, so
It's
malaria.
an Italian word.
Yes.
It comes from the Romans. Bad air.
Oh, they had
Malaria.
They had a city down in Italy called what was the name of that city, Dickson? It had Mal in it. Oh, it was wiped out by malaria in Roman times. Pestum.
I should know this, but I don't. I'm
Pestum is
sorry.
another
Maybe
word for disease.
some of our listeners will know. I know that they eliminated it from the port of Ostia during the reign of Claudius.
Okay.
How do you think they did that, Vince? They were building the port of Ostia.
Yes.
And in order to build a port, they had to fill in the swamp. And filling in the swamp...
That got rid of the standing water, right?
Yeah, but you see, the Romans at that point thought it was caused by bad air.
Malaria.
Malaria. Bad air. So if you can get rid of the bad air, you can get rid of malaria. Well, they were partially right. It was... if you got rid of where the bad air was coming from.
Right.
In this case, it was water, which was stagnant, which was supporting the growth and development of these mosquitoes. Okay, so Big Nami's group starts to investigate, just like Sir Ronald Ross did.
Correct.
They start by taking people now that are infected with malaria, that they can tell that they're infected because they're using the Romanowski stains. And by the way...
what do these stains do, by the way?
They differentiated between the nucleus and the cytoplasm.
Of the malaria.
Of any cell.
Okay.
Any cell. So it allows them to do slide work.
So they take a little bit of your blood.
Yep. They smear it out.
And they can stain it.
And they stain it, and they look under a microscope, and they can see something inside the red cells. Because all the red cells in humans don't have nuclei.
Right.
Which do, by the way. Do you know any animal groups that have nuclei in their...
Chickens.
Yes.
I used to work on chicken nuclei from red blood cells.
See? And amphibians do also, by the way. But not people. At least not in mature red cells.
Not the mature ones, but the precursors have nuclei.
Reticulocytes have something similar, but that's okay. That's a different story.
So they could see that there was something odd about these bloods. They didn't have the normal red blood cell.
But it's easier to tell than that. They just asked them, how often have you had these fevers? So they went back to Hippocrates. The Hippocrates actually classified the fevers from malaria as a 3-day or a 4-day long fever.
No, I don't get this. There are different length fevers you get?
There are, depending on the species.
In a particular sequence?
There are. Exactly right, Vince.
Okay, you're going to tell us at some point?
Eventually. Okay. We'll get to the clinical stuff.
Good.
But you want to know about the discovery.
Yeah. No,
and the controversies.
I'm sorry if I go all over the place.
No, you're not. I'm trying to bring you back. Okay. So here we come back to the main point, that here is a discovery that says malaria is transmitted from animal to animal by vectors that are invertebrates and, in this case, mosquitoes. Okay. Now, we already had one example of that with a tick, with a Babesia organism that looked like malaria. But ticks don't do it there. Maybe another kind of invertebrate does it here, and eventually Ronald Ross got it right. So now, how do you build on that result? You get an infected human, and you allow different species of mosquitoes to feed on them, right? Mm-hmm. And then you take those mosquitoes, and you see whether or not they can, A, transmit it to another person, or whether they have the stages of the mosquito, of the malaria parasite inside them. So that's not what they did first. You know what they did first? You can actually dissect a mosquito and look inside to see whether it has malaria. Is right. Thank you for that.
That's hard.
Not actually. If you turn the pages...
We're looking at Dickson's textbook here.
Keep going.
I'm going.
Stay right here. Here's the picture.
Sporozoites of malaria in infected mosquito stomach preparation.
Correct. A mosquito stomach preparation. So all you have to do is catch a female mosquito. Now... why do you think I said female? Because I'm a chauvinist, right?
No. Probably those are the ones that transmit it.
Why don't the males transmit it, Vince?
I think they die right away, right?
Well, just like us, after mating, they die.
They mating.
Bum bum. No.
Short lifespan.
Keep talking.
Something to do with a blood meal?
Exactly. Why don't you think male mosquitoes bite us? Because they don't.
They don't bite us?
They don't. In fact,
they hatch, mate, and die.
Something like that. Or they hatch, mate, and go off to suck on plant juices, which is what they eat.
They don't eat blood.
They do not. And in fact, neither does the female mosquito. She doesn't feed on blood either.
Mm-hmm.
She uses the protein in your red cells to make eggs. And since males don't make eggs,
they don't need blood.
They don't need any blood. So it's only the female mosquitoes that actually present a problem to us. All right. So Big Nami's group, okay, go off into the field, and they take all of these mosquito species, and they start looking. And they look, and they look, and they look. And they've exhausted all of the culisine mosquitoes they can find in the entire area around Rome. And they don't find a single infected mosquito.
Although we know there are people around Rome with malaria.
Tons of them.
Right,
in fact, if you read this book by Henry Miller.
Ah, yes. What was the name of it?
The book is called... Daisy Miller. It's by Henry James. I'm sorry, I got the name wrong.
Mm-hmm.
The author is Henry James, and he wrote a book called Daisy Miller. And Daisy Miller is about an American girl who comes to visit her relatives in Rome. And they tell her not to walk out at night around the Colosseum, and of course she does, and then she ends up dying.
Malaria.
From malaria. So malaria was really prevalent in that area. Mm-hmm. Tons of it. But they couldn't prove that the culisine mosquitoes transmitted malaria to humans.
Maybe
you know why?
It wasn't the culisine mosquitoes.
Well, that's exactly the story that they came up with. They said then Serrano Ross was wrong.
Well, he's right about canaries, maybe.
Now this sort of tells us about the host specificity of things, doesn't it? Bird malarias can infect people, and people malarias can infect birds. And each host has a different species of mosquito that transmits it.
You know, Dickson, I was beginning to suspect this. Do you know why?
I don't know.
This first plate in your chapter here, I was looking at this mosquito, and it's tilted as it's biting on the skin.
So it is.
But you said the culisines were parallel.
They are.
So I said, this either means that Dickson is wrong, which we know can't be possible,
exactly right.
Or that other species also will transmit it.
Right. So what did you think they did next? Well, they threw up their hands in despair.
The Italians? They went to eat, and that was it.
After they came back from dinner, they said, I guess we'll just have to use the Anopheline mosquitoes now. Because the culisines didn't pan out at all. So we'll just have to use... And son of a gun...
Bingo, right off the bat, they got malaria in the stomach preparations.
And from letting them feed on infected individuals.
Right.
And then they could transmit the disease from person.
But could they catch wild Anopheles mosquitoes?
It's harder to do that, but you can do it. You use CO2 traps. And they didn't do that.
Well, back then they didn't have...
No, you know what they did? They let someone stand outside at night. And just the mosquitoes, and then they just pick them off the individuals. So we do attract mosquitoes to us, by the way. It's not just a random event.
Of course. And what does attract them, Dickson?
It's a combination of many things, but the first thing is carbon dioxide. When we breathe out, it sinks to the ground and travels along the ground, forming a gradient, and the mosquitoes are lying in the grass, waiting for us to breathe. And they detect this signal, which tells them to migrate, and they fly in a zigzag fashion until they home in on the signal, and then they zoom towards it. Then they detect the presence of body heat. And I'm going like this to my eyes, but actually there are two large bulbous connectors to their antennae, and inside the bulb is an organ called Johnson's organ, and inside the organ there's the detector for body heat. And there it is, right there. And then not only is CO2 being given off, but it's from a warm animal. So they go and then they start to sniff. Now, some of us smell better than others to mosquitoes, and we know this. You can sit next to somebody outside, and you'll be drinking your mint julep, and they'll be drinking their scotch and soda, and they're covered with mosquitoes, and you don't have a single one on you. And you wonder, what the heck is the difference between me and them? And the answer is that this individual had a better smell than you.
Better meaning something that
it was more acceptable to the mosquito.
We
more
don't
desirable.
We don't know what that constitutes, though, do we?
We don't, but we've got a guy here who's got a Nobel Prize for his work.
I know who that is.
I bet you do. Tell the audience.
Richard Axel.
That's correct.
Who discovered the receptors for scent in their nose.
And what do you think he's working on now?
It's mosquito scent.
He is.
Yeah.
He is. If you could interfere with it, then you could prevent the mosquito from finding you. Like a
so
smog.
They must also be able to distinguish different species of mammals as well.
They can. They have biting. They have feeding preferences.
Yeah.
But if something isn't available to them, they will substitute another. So it's preferences. It's not rigorous. Okay. So the point is that the Italian group got it right for the transmission of malaria to humans.
Mm-hmm.
They discovered anopheline mosquitoes transmit.
The Italians did this.
They did. And the anopheline mosquitoes have a totally different pattern of lifestyle than the culisine mosquitoes.
How so?
Well, they prefer different kinds of water. Some breed underneath trees and shade. Some prefer warm. Some prefer cold. But they mostly prefer clean water, whereas culisine mosquitoes love dirty water. That's a big difference. Now, so here you are in Sweden, set to give out the Nobel Prizes. And who do you give them to? Because you can only give them to 3 people, right?
Well, there was
give one to Sir Ronald Ross,
sure.
give one to Lavran. And you'd think, of course, that Bignami would be a shoo-in.
No.
Not even considered.
Why?
Don't look at me.
Derivative.
Maybe.
It's a mosquito. It's just a different species.
Yeah,
you know?
But what a difference. People are different. So here you had some bone of contention that arose after the prizes, of course.
Who was the third?
It's a good question.
Nothing to do with malaria.
No, it had something to do with malaria. I'm just blocking on their name right now. We could probably go to the internet and find out.
The what?
The internet.
So
I think it was 1907, but I'm not sure about the exact year. You just type out Nobel Prizes, and you can go back to the very beginning of Nobel Prizes, right?
1902 Nobel Prize was awarded for the discovery of... I'm waiting for my page to load here. The discovery of... And now I ended up somewhere else. I ended up at the main page.
Right.
Oh, here we go. For the discovery of the parasite causing malaria. And the 1907 was for finding the parasite in human blood.
So
let's look
at...
Who
got it?
Who was 07? 02 was Ross.
Yeah.
By himself.
07 was Lavran. No, Lavran worked with birds, so he got it for discovering
and in
the
07, it was Lavran.
Lavran, okay. Frenchman working in North
Africa. So
Africa.
it's only 2.
Frenchman working in North Africa. Rode his bicycle from the place of collection to the laboratory, and in the meantime, in the little blood sample, the parasites underwent their developmental stages. By the time he got back to the laboratory, they were visible underneath the microscope. There's a rich history of work. Knowing that it's transmitted by anopheline mosquitoes allows you to target control mechanisms against the mosquito, right? So these are night-biting mosquitoes mostly. Culisine mosquitoes bite many times during the day. Many species of culisine mosquitoes are day feeders. Some are night feeders, but many are day feeders. So those would be the ones that you didn't have to worry about, okay? It was at night that you had to worry. And that's where Henry James picked up on that idea and decided to write a story about it.
Sure.
And it was a story of manners and stuff like this, too. It's kind of a... I guess it's a highly structured story. I don't want to denigrate the story much because I think it's a brilliant writer. Henry James is a brilliant writer. And I've read this story many times. And... but you have to get used to the syntax of the writing in order to find an appreciation for it.
So was that an important discovery for control? Because at that point forward, you could start reducing mosquito populations?
Sure.
Were means available to do that?
Well, there was a single drug that proved to be the turning point in all of this, and you can probably guess the name of that drug.
I think it contains your initials, doesn't it?
No. That's to combat the mosquito.
That's what
How about the parasite?
Yeah, chloroquine.
No, that's a modern derivative. I'll give you the hint. You take some gin, and you take some tonic, you take some ice, and you take lots of lime juice, and very little tonic and lots of gin, and you get yourself a gin and tonic, right? So gin and tonics, this was the way. Now, what is in tonic anyway?
Some herb stuff, right?
Yeah, well, tell me about it. Do you know the name of it? Yes, you do. Quinine.
Quinine. Quinine is really, even in contemporary tonic, there's quinine.
Where does quinine come from?
The quinola plant.
No. The cinchona tree. The bark.
It's close.
No, you weren't even close, Vince. I won't give you an A on that one. I'll give you a D- for not knowing that the Peruvian princess, as a gift, gave to the conquistadores the secret to malaria control in Peru. And you know what they did? A, they took this nut of the tree back and grew it in Spain. B, they killed the princess. And C, they took all the gold.
She knew this was a preventative for malaria?
They all did. Yeah, the Peruvians already knew this. The Incas knew this already.
Amazing.
If you take the bark of that tree, just like the Native Americans knew that if you took the bark of the willow tree, the white willow, that you could extract treatment for headaches, right? Which we now call aspirin or acetylsalicylic acid.
Right.
In the case of the quinine, the cinchona tree had the active ingredient, the bark, and by crushing it up and making an extract of this, it was so bitter, though, that no one could stand a full dose. You had to cut it with something. So the British, of course, worked all this out rather well, you know. So I just have a little bit of gin and a lot of tonic, and I say, you know, you could probably end up still catching malaria because I don't think you can achieve therapeutic levels, but you won't actually care because if you use enough gin, of course.
The British way, but the Peruvian way. So this would be given to people who had the fevers of malaria?
Oh, yeah.
Is that the idea?
Oh, absolutely.
And would it help them?
It cured them. It didn't just help them. It cured them of their blood stages.
But what you're saying in a gin and tonic, you can't get enough of the
Right,
active ingredient
that's
to do
right,
any
that's right.
good. Fabulous.
But it was the
This
origin.
is natural products.
It was the first wonderful use of a natural product. And do you know the name of the company that was established to produce quinine? Sure you do. It was the Burroughs Wellcome.
Is that right?
They made
I didn't know that.
gazillions amount of money on
They
quinine.
still do.
And they made so much that they split off into two companies.
They have a foundation, right?
That's right. The Wellcome Trust
Yeah.
probably has more money than the Howard Hughes, which has a huge amount, right?
I need to write a grant to support these podcasts.
And all of that, right? If you're listening out there, anybody with a lot of money, just send it right to us, and we'll be glad to continue this conversation.
Dixon, so the nut that the Peruvian princess gave to
no,
the
she
Spaniard.
gave them the bark. Gave them the bark. They discovered it came from a tree. Then
she didn't give them the nut.
No.
Ah,
She gave them the bark.
okay. And they took the seeds back to Spain and
did.
eventually propagated throughout Europe.
That's correct.
And of course, the fortunate part is that the tree grew there under those climate conditions, so
absolutely.
that's fine.
Absolutely.
And they used it to treat malaria for
There you
many
go.
years. And did we get resistance very quickly on?
Not very quickly, no. Not very quickly at all, in fact. In fact, if it was used therapeutically. Okay.
But nowadays we don't use quinine.
It was later on, of course, the chemical industry. Do you know which drug led to the establishment of the chemical industry? The pharmaceutical chemical industry was established by one company.
Is that Bayer?
How did you guess that, Vincent? You are brilliant.
No, I just
A
guessed.
plus. That's an A plus.
I don't
And
know
which drug
why.
did they make?
Aspirin?
Yes.
Bayer aspirin.
Yes.
And that established the whole industry. What year would that be?
It was in 1890. 1890. It was in the 1890s. And you know who Friedrich Wöhler was? Yeah, you do, but you just don't remember. He was I believe he was a German scientist who synthesized the first organic compound. Do you know which compound that was?
What was that?
It was urea.
Okay.
Which turned out to be a prime ingredient in plant fertilizers. All right? So the beginnings of the pharmaceutical industry and the beginnings of the chemical synthesis industry occurred in the 1890s. By the time quinine was discovered and then it was synthesized.
Yeah, they figured out what it was and they could make it, yeah.
And then when resistance did occur, or when the therapeutic doses were so high that you had to give them a lot orally, people in a coma could not be given quinine because some of these malarial species will cause you to go into a coma, particularly Plasmodium falciparum. We haven't even discussed the species yet.
No, we haven't.
We haven't, but this is some of the history of the establishment of the field of malariology as it is known today. Okay, so here we have the rising of the chemical industry, the arising of the pharmaceutical industry in the 1890s, the discovery of vector-borne diseases in the 1890s, the discovery that mosquitoes transmit diseases in the 1890s. Now we have a whole bunch of things coming together, right?
And when did the quinine become...
Oh, that was in the 1600s and 1500s.
So that was just for many years to treat malaria.
Yeah.
It had nothing...
Before they even knew what caused it.
So the discovery of the mosquito vector had no impact on that, zero.
None.
They continued to use it into the 1900s, I presume.
Today, they're still using it today.
Mm-hmm.
And there's a derivative of it called
Quinacrin.
Quinidine, which is the safer drug of a quinine derivative.
All right. But this discovery of the mosquito transmission...
Yes.
How was that useful?
Well, it wasn't immediately useful, although you could sleep underneath mosquito netting and keep them away from you. If you knew to avoid mosquitoes, you could avoid the infections. Eventually, a compound was discovered which could be used against the mosquitoes. Can you guess the name of that compound?
Is that the one with your initials in it?
That's the one.
DDT.
That's right. What does that stand for?
Well, I got to look it up. Hold on. I want to look it up because this is how we learn. We type DDT.
That's right.
Well, the trivial name is diphenyl trichloroethane.
Correct.
It's one of the most well-known synthetic pesticides.
Here. And who discovered that, do you think?
The Swiss chemist Paul Hermann Müller awarded the Nobel Prize in 1948 for discovery of DDT.
How do you think he discovered it?
Hmm.
Yeah, you don't just discover DDT, do you? But actually, you do.
Well, it might have been present somewhere.
It was.
Was it a natural compound?
No. It was a synthesized compound of the Bayer Company. And it sat on a shelf.
Yeah, of course.
And what they did was, of course, went down that shelf list of chemicals and tried everything against malaria. The parasite, in this case, they were trying to kill off the mosquito. And this one worked. Now, when do you think they discovered that? He got the Nobel Prize in 1948. That's after the Second World War.
Right. 1939, Vince, it says here.
That's right. What's so propitious about the date 1939? This guy was Swiss, right? He was Swiss?
Yeah, he was Swiss.
But the compound was made by Bayer. It's a German company. 1939 is a bad year to be in Europe.
Yeah.
It's the beginning of the Second World War.
Yes, that's correct.
What happened to that compound?
In 1939?
Yeah. It was known to have anti-invertebrate activity.
Mm-hmm.
I mean, it kills a lot of different things, not just mosquitoes. It kills almost all invertebrates. It's a neurotoxin. It's an organophosphate. Okay,
right.
So the point of this whole exercise is that this was snuck out of Europe, and we came into possession of it, and we used it.
It says here it was first made in 1874, so it sat on the shelf until the 30s.
Right.
But I'd like to know who had the first hint that it was a arthropod compound.
I think the Swiss chemist.
Why did he get it?
Good question. Probably because he was doing some investigations on maybe another arthropod-borne infection.
Typhus.
There you go.
Which was a big problem
there you go.
And had been for centuries, right?
Right. It defeated Napoleon.
Yeah.
And you know what else it did? It defeated the Germans and the Italians during the invasion of Italy by the United States and Allied troops.
Really?
How did we survive that, do you think?
Did we have DDT?
We did. And they flitted us down the back with these big canisters filled with DDT, and our bodies were completely lice-free. And meanwhile, the Germans and
our
the
eggs
Italians,
had soft shells then.
The Italians were dying off like
flies. Is that
flies.
right? I didn't know that because I read that book, Rats, Lice, and History.
Right.
By Hans Zinser. And I don't think he talked about, because it was written probably in the middle of World War II, so he didn't have the story yet.
That's exactly right. Exactly right. And so when we invaded Naples, if we had not protected us from lice, we would have all died from typhus.
Typhus. Huh?
That's right. So let me go back to malaria, because the typhus is a vector-borne disease too.
Yes.
Okay, so now we have...
And the agent of that is Dickson? The agent of typhus?
It's a rickettsia.
Exactly.
It's Tsutsugamuchi.
Yeah, I'd say that because in the book...
No, it's rickettsia. That's the name of the organism.
In Rats, Lice, and History, you keep saying it's a virus.
No, it's not a virus.
It's not a virus. Yeah.
It's an almost bacteria.
That's right. Okay.
So So malaria has played a huge role in history, throughout history. I'm glad you brought up history.
Tell us.
I will tell you. Let's just take the Second World War as an example. I mean, I'm old enough to remember a portion of it because I was born in 1940. So as a kid, when I was 5 years old, the war was still going on. How do you think it ended? Badly for both sides, trust me. I actually heard on the radio a broadcast of the atomic bomb.
Yeah, the Pacific theater, yeah.
Yeah.
But the European theater.
So the
ended in
atomic bomb
different ways.
killed many thousands of innocent people,
Sure.
didn't it?
And hundreds of thousands, yeah.
It was nothing compared to the number of innocent people and not so innocent people killed by malaria during the Second World War. More people died on both sides of the equation from malaria than from bullets. Isn't that amazing?
Is this because there were massive movements and exposure to the mosquitoes? Not so you wouldn't, in a normal life without a war, you wouldn't have so much mortality?
That's correct. Or you wouldn't have so many people crowded into some small spaces.
Mm-hmm.
Like, for instance, in the First World War, for the transmission of influenza virus.
Right.
Same deal. When you crowd people together, that's a perfect setting for a parasitic infection to take off. So here we had all these troops crowded into similar small areas. They were given a drug to take,
Mm-hmm.
right? And what was that drug, do you think? It was a derivative of quinine.
Okay.
It
Was a
called,
derivative of quinine.
what do you think it was called?
Quinacrine?
No. Adabrin.
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 Okay, I've heard
And
that
it left a horrible taste in your mouth. It was
Mm-hmm.
bitter as hell. And it took away your appetite. And it gave you incredibly vivid and perhaps some bizarre dreams.
Oh, I know about that. Yes, my student who went to Africa once told me about those dreams.
Right. So most of the quinine derivative drugs do that to you.
Mm-hmm.
But being shot at, I think, is another good reason to have a bizarre dream, don't you think?
Yes.
And so these troops would actually stop taking the drug. Okay? When they did, of course, they would acquire their malaria. How did they know if you were or were not taking Adabrin?
How would someone else know?
How would they know?
You had fevers.
Nah, there's another way to tell here. This drug has a multiple anti-parasitic effect.
Mm-hmm.
It also is effective, believe it or not, against another protozoan infection called Giardia. And a way to tell whether or not you were lying about taking your Adabrin. Oh, I've taken my Adabrin religiously. I did. Really? Well, come on in the laboratory, and all I need is a stool sample from you. And if I don't find Giardia, then I know that you're not telling a lie. But if I find Giardia in your stool, then you're telling me a lie, because Adabrin also cures Giardia.
Yeah, great.
You know what else it cures?
What else?
Even more amazing. It cures adult tapeworm infections.
Is that right?
That's right. Exactly right, Vince.
Adabrin, yeah.
One drug.
Right. It kills the tapeworm.
It's amazing.
It dies within you, but you don't know if it's gone because it degrades inside you.
Because your digestive tract takes
Yes,
care of
I
it.
remember
That's right.
we spoke about this. That's
did.
why in some cases they put a tube in and
We did.
put the drug right on the worm.
Well, that was another drug called aspidium. That's derived from a male fern plant. I think we're confusing aspidium with Adabrin.
But the woman who put the 7 worms out,
Yeah.
they had put a tube in her and
They did.
deposited ethidium. What's the drug? That's a stain for DNA.
Aspidium.
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 That's what they put in. And then Adabrin you would take, and it would.
That's
Because
right.
actually we have an email from someone who
Yes.
asked, "How do you treat tapeworm infections?"
Well, there are many drugs now available.
So Dickson, what do you want to do here? Is this a good point to stop?
It is, because now we can jump into the organism
All right.
next, and I can tell
Want
you
to read a
some
few
stuff.
emails?
Okay.
We always
sure.
have good ones.
Hey, come on, bring them on.
Email number 1 is from James, who's from New Zealand.
Oh, yeah. My favorite country.
One of the other science-based podcasts I love listening to is an actual radio show in New Zealand called Our Changing World.
Huh.
Notice that. An actual radio show.
As opposed to an ersatz radio show like
is
ours.
ersatz. That's right. This week, the final story was on hookworms.
Uh-huh.
And some researchers studying it, looking for a vaccine, and seeing if they could take out the anti-inflammatory ability of the hookworm and turn it into a drug or treatment without the downside of having to be infected by the worm
Wouldn't that be
itself.
cool? That'd be cool.
I thought you guys and the listeners would enjoy it, and it's a short 22 minutes. So he sent us a couple of links
Nice.
to the website and to the podcast versions.
Does he mention who the investigators are?
It doesn't,
Okay.
but we'll post it and everyone can
Because
look.
one of the authors on this book that we've written together is Peter Hotez, and he's a world leader in
Is that right?
a vaccine developing against hookworm.
Let's see if on the webpage they have the name of any of this.
Okay.
We'll just check very quickly.
I don't know any.
Radio New Zealand. No, it's not obvious here, and I
Okay.
won't spend the time. But
We'll have to listen to it to find out.
he ends by saying, and before I forget, insert platitudes on how awesome TWiP is and how it needs to become a daily show here.
A daily show? No, come on, that's pressure, baby. We need to get paid for this. Then we just have to. I mean, just slip the food under the door, maybe, or something. Who knows?
Under the right conditions. Thank you, James. Peter writes, one of your emails mentioned larval stage parasites.
Yes.
If you consider placental mammal gestation, I think the embryo and developing offspring in utero fulfills many of the criteria of a parasite.
I would agree.
It basically plugs in and takes everything it needs from the host mother until it reaches the next stage.
This is correct.
An intelligent egg-laying, say Martian, species may well view this as a hideous form of parasitism. You agree?
Not hideous, but it certainly is a form of parasitism.
Well,
there's no
a
question.
Martian might view it as
That's right.
hideous. Yeah.
But there are some selective advantages of placental development, so that you can say that you don't have to worry about a cat stealing your eggs and crushing all your offspring.
But it doesn't spend its whole life as a parasite, although you might, having had children, you might think otherwise.
They never stop being children, that's for sure.
But do true parasites, the kind we talk about, aren't they parasites their entire life cycle?
No, some of them have free-living
these
stages. Some of them have free-living stages.
That's true. But you would say technically a human could be a parasite.
Not just technically.
I'm
saying the lifestyle of an embryo is a parasitic lifestyle with regards to the human.
Okay.
I would.
Thank you, Peter. Nick writes, first off, when I hear your ending song and thought it sounded like Ronald Jenkins and just saw it was, I like his stuff. All right, good. It is Ronald Jenkins.
Right.
And the beginning song is too. So I heard on this American Life podcast last week a story about a guy who heard about hookworms having an effect on whether a person has asthma or allergies.
Correct.
So he went to Africa and walked around in the poop fields and got hookworm. When he gets home, his allergies are gone. What do you think about that? I have allergies to cats. Should I go get some worms? This was also first on a podcast of WNYC's Radio Lab, which I highly recommend. Hey, Nick, Dickson was on that podcast. That was
true.
Dickson Despommier, who was sitting here.
That was more.
And you and Carl Zimmer
Yeah.
did that episode. So should he go walk and poop?
No. I think cat allergies are minor details, minor problems. I think if he had Crohn's disease, I would recommend another parasite. It's called whipworm, not hookworm, whipworm, Trichuris.
Mm-hmm.
And that is being used therapeutically now with IRB approval for giving a person a short-term worm infection to abate the symptoms of Crohn's disease.
Why don't they purify the protein that does it?
Tell me, because of the lack of financial support from a central governmental funding agency such as NIH.
But Crohn's is a serious issue.
It's very serious. You can die from it. So someone should be willing to fund this kind of research. I think there's a Crohn's Association. If you could identify the target that the immune system points itself at from the parasite and synthesize it, you could keep it away from the gut tract and keep the host from killing itself. Yeah, that's a proven scenario.
Jim sent us a link to a story, tapeworm brain infection, serious health concern. Tapeworm infections of the brain, which can cause epileptic seizures,
Correct.
appear to be increasing in Mexico and bordering southwestern states.
This is correct.
Loyola University Health System researchers report.
Yep.
Does that make sense?
It does, and it is increasing.
All right, we'll post that story. Thanks, Jim. Why is it increasing?
I think because public health meat inspection systems are breaking down and more homegrown produce is being made and more recycling is being encouraged, which sometimes encourages this cycle to continue, like using human feces as fertilizer.
So this is the problem where you eat the eggs of the pork tapeworm and then it goes and burrows somewhere in you.
That's correct, and you get cysticercosis.
Right, neurocysticercosis.
That's correct.
Brian writes, I heard that every vertebrate has its own tapeworm, but what is the human tapeworm? In other words, if a tiger eats a human, what tapeworm does the tiger get?
None.
No, there are none that are transmitted that way. You know why, Dickson?
Tell me why.
Not enough people get eaten by tigers. It hasn't evolved, right? But maybe
correct.
Back when hominids were walking around, it might have happened.
If you go to the Sundarban in India, people are regularly eaten by tigers.
Oh, yeah?
Oh, yeah.
But it's not a sustainable activity.
No.
It's not evolutionarily sustainable. You know what I'm talking about.
I do
right?
know what you're talking about. Yeah.
Thanks. P.S. Now I want to find some fake gravid proglottids and leave them in my roommate's bed.
Don't do that.
Oh, that's not nice.
No, that's not nice at all.
Thanks, Brian. Gopal writes, I saw this piece in the New York Times and remembered Dick talking about the life cycle of the pig tapeworm. This is from Gopal, who's a science journalist in India and a regular listener of both TWiP and TWiV.
Great.
The article was Global Update Kenya Pig Farmers are focus of effort to stop spread of parasite that causes epilepsy. Sounds like a similar story to what Jim sent us. Yes.
Yep. And remember the original one that was in Papua New Guinea. That's where that's where the idea first occurred to us.
Arson writes, hey, you talking about fish tapeworm reminded me of another interesting fish parasite. Oh, have you seen this? This is a parasite, eats the fish tongue and replaces it. Have you heard of this, Dickson? I'll show you the picture. It's called a tree hugger. Keep up the good work, Arson. I saw some of these photos, and here is this parasite that eats the fish tongue, and then it sits in here in place of the tongue. Okay.
What the heck?
You never saw this? It's a sea-dwelling parasite, attacks fish, burrows into it, and then devours its tongue. After eating the tongue, the parasite proceeds to live inside the fish's mouth. And the fish is okay, and this guy
looks like a
gets food.
Crustacean of some sort.
It does, doesn't it? I'm not sure this is real.
It looks more like an alien story to me.
I think it's somebody who stuck a crustacean, or found a fish with a crustacean and it took a picture of it.
I must tell you that, by the way, this is a total aside. I was watching the new movie Avatar on the small screen on my way back from the Canary Islands, and I was totally disappointed with the whole deal. It's just the biology in there, so...
The biology is not so great, but
It's
if you see it on a big screen, it's quite amazing.
I
production.
guess.
Here's
I
a
guess.
side view picture of this...
Critter?
thing. Yeah, see it's in there. I think that was just something that washed up on the beach, Dixon.
I want to see the life cycle of it.
You know what's disturbing? This article has had 7,992 hits.
Well, it's a bizarre thing. People are interested in the unusual.
Thanks, Arson.
Parasites are very unusual.
Parasites.
Sure.
That's right.
They're more bizarre and they're the truth.
Listen to this one, Dixon.
Sure.
Sky writes,
Okay.
just listen to the last episode, TWiP 7, tapeworms are fantastic.
Okay.
And you know what? You both are fantastic, almost like the tapeworms.
Hey.
That's pretty nice.
Well, I'm not
fantastic. No.
I'm not fantastic like a tapeworm, I hope.
I'm glad you enjoy the teaching, but I'm personally not fantastic.
Yeah, well.
Would you consider yourself?
Of course
No.
not. Of course not.
Brent writes, I love the podcast. You've been talking about tapeworms. Can you talk more about the treatment for tapeworms? How does the treatment work? Does it kill the tapeworm or just force it out of the body? Are there any issues or side effects with the treatment? So you can do either one. You can kill it in the body, and what's the name of that drug?
Praziquantel?
Or you can put the drug right on it, which requires intubation.
No, we don't do that anymore. Praziquantel is a drug of choice.
So it kills it in situ.
Yeah.
And then you digest it.
Yes.
Side effects? Very little.
Almost none.
And that's the treatment for tapeworms.
Yep.
Well, maybe when we'll have Josh Stillman on, he'll confirm.
Sure.
Or deny.
Or deny.
All right, we have just two more, and then we'll finish.
Okay.
Michael writes, I'm a medieval history student who became fascinated with parasites from dealing with the plague. From there, I developed a course on plagues and people through history, which covered both parasitic and viral plagues, which have wiped out large portions of humanity throughout history and their effect. I have fell in love with both TWiP and TWiV. Thanks for taking the time to these... Thanks for taking the time to something school and has been more episodes. Something's missing here. I'm sorry, Michael. I've finally gotten a copy of Dick's most recent edition of the book, and it is my bedtime reading. This is Dr. Michael.
Well, Dr. Michael, I hope I'm not keeping you up at night.
One more from...
Oh, I wanted to say something here
Sorry.
because
Yes.
the plague connects me back to another something that I've been reading myself. It's a book called Archimedes, From Archimedes to Hawking.
Oh.
It's a wonderful read about the laws of physics. And one of the stories is about Sir Isaac Newton. And Sir Isaac Newton was forced out of Cambridge in 1666 by none other than the Black Death, plague. And you know how they tried to control it? Because they didn't know how it was transmitted. They didn't have a clue that it was vector-borne, you see.
Right.
But they suspected, because of all the stray dogs and cats, that it was either from stray dogs or stray cats. So you know what they did? They killed all of the stray dogs, and they killed all of the stray cats. And guess what the stray dogs and the stray cats were eating? Rats. So the rats actually increased in numbers, and in 1867 the plague got worse than it was in 1666.
Bad.
You know how many people were dying per month from plague?
No.
1,000 people per month. Isaac Newton went to the countryside to escape it.
Yeah.
How do you know that that's the way to escape it? But that's what they did, and that's where he came up with all of his wonderful laws, including the gravitational.
Well, that's a good side effect of him fleeing.
Fleeing, there's no pun intended there. You can't flee from the plague.
Now you're taking the part of Alan
No
Dunn.
matter how mighty you trust your feet, you cannot flee from the plague.
One more.
Please.
Then Dickson has to go teach in my virology course.
Okay.
Kaja writes, I think it's Kaja, K-A-J-A, from Slovenia. So, dear TWiP, I've been following you from TWiV, and although I'm more of a virologist, I have to say TWiP is really amazing. I enjoy Dickson's way of narrating these amazing life cycles of parasites. It really is an exciting subject. I even looked up study programs of parasitology at our university. As you pointed out, it's quite similar to virology on a big scale. My question for Dick is, how does the life cycle of a parasite even occur? Did the tapeworm exist in some form before it became a parasite, or was it just a parasite in one organism before it evolved to transform and change hosts in different stages of life? Did the evolution somehow make an opportunity for its life cycle in dog, sheep, dog? Surely dogs weren't always around sheep to catch the worm, and thus giving it a chance to evolve in this sophisticated life cycle with so many different forms of the worm. I hope you understood the question, and maybe in one show can explain the evolution of parasites. This podcast really is amazing to learn from, and I wish you would make it more often. I really appreciate all you work with TWiV and TWiP. It's a pleasure to be taught by you. Keep up the good work. Greetings from Slovenia. That's a whole show in itself, right?
It is. Of course it is.
And in fact, I alluded to it earlier.
You did.
Because it's a fascinating subject.
How do these things arise? I think life is always trying to break into the safe. We are the safe. You know, they had a contest once at Science Magazine to see who could come up with the most profound statement of discovery over the last 100 years. And you'd think it would relate to something like Darwinism or something like that. But the winning slogan was, successful systems attract parasites.
Yes, of course. I love it. That's our subtitle here
for
this
It's the best.
podcast.
So we are one of the most successful systems on Earth. Why? Because we cover the Earth with ourselves. Our species is spread everywhere. We eat everything, and we drink everything. If we do those two things, it's for sure we will encounter virtually every living microbe in the planet and on the planet. And many of them will have a chance to try to crack the safe and to enter us and to take advantage of our successful system. So it's a constant struggle, and we know this. It's like an arms race war, right, between the immune system of us and the potential parasitic organisms outside, trying to gain an advantage by being part of us and to actually have us feed them. So it's amazing that we aren't infected with more things rather than less. I mean, if you look at how many organisms actually have succeeded at colonizing the human host, and then ask how many different kind of organisms are there, you can see that there's quite a big difference between how many organisms there are versus how many that have succeeded. And so I think it's a very small fraction of organisms that actually find a niche inside of our body someplace and successfully complete their own lives in a way which resembled another life cycle that they were part of prior to that. One builds on the other. So there are some wonderful books on this subject. Nobody has a definitive answer, by the way, so these are all speculations. But it's just a series of trials and errors as organisms and hosts react against each other and play off one another and develop habits that reinforce one or the other. When we talk about toxoplasmosis, the listeners will be absolutely stunned to realize that if a mouse or rat catches toxoplasmosis, they actually lose their fear of cats.
Yes. Wonderful story.
That's a fabulous story because at that point you are, as they put it, dead meat.
But it does it happens more slowly than with viruses. That's a key.
Sure, because the replication is much slower.
The error rate is slower.
That's right.
But
you'll see that one of the species of malaria will be an exception to that rule, Vince, and we'll talk about that because it has a very rapid rate of developing resistance to drugs.
Well, I'm sure at some future episode we'll deal with this some more.
Yep.
There are people who work on this whole issue.
We probably have to have them on our show.
I think we should.
Like Tom Wellams down at NIH, he would be a fabulous guest to have on our show.
Yeah, I think we should do that. Absolutely.
Sure.
All right, that'll do it for today. Thanks for everyone for listening. This has been This Week in Parasitism. You can find us at microbeworld.org/TWiP. You can find us at iTunes, and if you do subscribe there, which would be great, leave a comment, just a short one. That helps us stay on the front page of the podcast and get more people to listen to us. Thank you, Dickson.
You're welcome, Vince.
Good to see you again.
Same here.
See you in 2 weeks.
Good to see you. Yep.
For TWiP and TWiV. Yes? What's the matter?
Come on, you have a tag for the show.
Oh, yeah, I was just saying goodbye. Glad to see you again. We missed you. You have been listening to This Week in Parasitism, the podcast, and I think the only podcast, all about eukaryotic parasites. Thanks for listening, everyone. We'll be back soon. Another TWiP is parasitic.